Uncovering the secrets of the adult brain. Brain cells continue to develop even in adulthood. Forget the myth that you stop growing new brain cells once you reach adulthood. Recent research has cast this outdated belief aside by demonstrating that our brains keep growing new cells well into later life. The study, spearheaded by Swedish scientists and published in Science on Thursday, highlights their discovery of active neural stem cells in the hippocampus of adult brains. These findings not only offer new insights into how our brain changes as we age, but also open doors to novel treatments for neurological conditions. According to Marta Paterlini, one of the study's co-authors and a neuroscientist at the Karolinska Institute, we've gathered solid proof that the human brain produces new neurons throughout adulthood. This helps to clarify that new nerve cells can indeed develop over time, marking a significant shift from earlier beliefs that adult neurogenesis does not occur. But this issue is far from settled. In fact, earlier studies have presented conflicting views. Some argue that new neuron generation significantly tapers off with age. The contentiousness surrounding neurogenesis is still a hot topic in scientific circles. Just in the spring of 2018, two highly regarded research papers that examined adult neurogenesis came to opposing conclusions. In their study, the researchers set out to confirm one key aspect of adult neurogenesis: the ability to identify and track neural progenitor cells, essentially the cells that will develop into new neurons in the adult brain. Using cutting-edge techniques, they meticulously examined brain tissue samples from individuals aged 0 to 78. They identified over 400,000 individual cell nuclei, revealing neural progenitor cells at multiple growth stages, particularly in the dentate gyrus, a crucial area for memory formation within the hippocampus. Paterlini remarks, "We observe clusters of dividing precursor cells right alongside fully formed nerve cells, in the very places where research on animals has shown adult stem cells live." This strong evidence helps bring closure to the long-standing debate about the brain's ability to generate new neurons in adulthood. However, this breakthrough also raises new questions. For instance, the gene expression patterns of these adult precursor cells differ from those found in animals, posing intriguing possibilities about how human neurogenesis might be unique. Additionally, it appears that some adults have richer populations of these neural progenitor cells compared to others. This variability might offer clues about why some individuals are more prone to neurological or psychological conditions. Moreover, finding a way to safely enhance the adult brain's capacity to produce new neurons could lead to groundbreaking treatments for these conditions, or even better recovery following severe head injuries. Recognizing that our brains can constantly produce new neurons reshapes our understanding of lifelong learning, recovery, and the untapped potential of our brain's plasticity, concludes Paterlini. There's still a lot to uncover about how our brains adapt and evolve over time. The team is now setting their sights on exploring other possible neurogenesis sites in the adult brain, including the wall of the lateral ventricles and adjacent areas. One thing seems clear. For some individuals, new neurons continue to form and replace themselves well into adulthood. What are your thoughts on enhancing neurogenesis in adults? Could this be a breakthrough for treating neurological conditions or aiding in recovery from brain injuries? Share your perspective in the comments below.